Welcome, everybody. I am so pleased to see all of your faces. Thank you so much for joining for what will be a very special conversation entitled Fast Track to a Cure, Employing a Business Mindset to Accelerate Cancer Prevention Research, which will highlight the accelerated impact the RUNX1 Research Program is having in the rare disease space by adhering to a successful organizational model that takes an entrepreneurial, less traditional approach to doing business as a nonprofit. In a very short time, RRP has created the first ever research network and patient advocacy group for RUNX1 FPD. We have grown to include 1,300 basic scientists, translational scientists, physician scientists, genetic counselors, and most critically, over 360 patients. As many of you know, I am Alex Gonzalez. I'm the Director of Development of RRP, and I have the pleasure of moderating today's discussion, which will be in a fireside chat format. We really wanted to provide an opportunity for a more informal and intimate setting to discuss the Foundation's work and impact. At the end of the conversation, there will be some time for Q&A. You can put your questions in the chat or unmute and ask directly. Here with me today is Tim Babich, co-founder and director of RRP and CEO of Nexus Holdings, who joins us from London. I also have Dr. Katrin Ericson, executive director of RRP, who joins us from our home base in Santa Barbara. Tim, who is also a patient, and his wife, Monica, established RRP in 2016. Katrin came on board as the Foundation's first staff member in 2018 and oversees RRP's research portfolio, as well as leads all aspects of the organization. Without further ado, I'd love to begin our conversation. Tim, please give us some background on what motivated you and Monica to establish the RUNX1 Research Program.
Thank you, Alex. Thank you for that introduction. We decided to found RUNX1 really on the back of a long-standing desire to set up something to help families like ours to have a cure for RUNX1 before it progressed to AML. My mother, Kathleen, who's actually on the call here, was diagnosed with AML in 1995 when I was a freshman at college, and she had a bone marrow transplant in the spring of 1996 with her sister being a donor. A couple of years later, we were able to put two and two together, just at the sort of early stages of genetic sequencing, that this was something that was prevalent in our family. And so, it was always on the back of my mind that I wanted to do something about this. I didn't want to have things happen to me and my family, but I wanted to at least try my hardest to be able to find some sort of cure. And that was one of the motivations in business, was to be able to be in a financial position to do something about it.
Thank you. Absolutely. And kind of moving into that, you've been extremely successful in your career. How do you feel your professional background in finance influenced the organization from the very beginning?
Well, I think in a number of ways. First and foremost, I think that we are extremely goal-oriented as a foundation. We like to say, where do we want to get to? And then we work backwards to try to see what are the steps to actually get there. And so that's first and foremost. I think secondly, we like to introduce accountability in the scientific research process. We often ask for a decent amount of progress reports to ensure people are on track and that they're sort of following through what they said they would do. But then, I think just, that's sort of in running a business. I think also, I'd add that, we're very focused on collaboration and teamwork within the scientific community. I think that that's a successful hallmark of many businesses, and that's something that we really, not only do we encourage in the scientific community of RUNX1, but we have that in our grant contracts, where there's certain sharing of information and collaboration that's required of everybody who's a grantee. But I guess lastly, part of, I think, at least what's influenced me in my business career is being able to ask the right questions. And so we're always trying to figure out, are we asking the right questions? And are the scientists that we're funding asking the right questions to actually try to get to a cure? Because, we don't want to take our eye off the ball of trying to get to what our ultimate mission is, which is a cure.
Great. Thank you, Tim. Moving on, Katrin, you came to RRP in 2018. What drew you to join the organization, and how did you feel your background complemented the Babich's vision?
Thanks, Alex. Hi, everyone. Nice to see you. Well, if I may, Alex, I'll start with my background and then how I was drawn to working with the Babiches at RRP.
Sure.
So I went into science for a couple of reasons. One was because I heard about cancer early in my childhood. So my dad lost his father to lung cancer when he was a young adult, and it was very difficult for him. I knew that he missed him very much, and I felt disappointed that I never got to meet him because medicines couldn't save him. And then secondly, I was, truly fascinated by cancer cell biology and how one's own cell could go rogue, multiply, and then, ultimately take over the body and kill a person. And so this was really a problem I knew I wanted to work on. So I joined the UCLA graduate program after undergraduate, and I was planning on conducting cancer research, but I ended up joining a developmental biology lab where I learned how the blood system forms in utero, and then actually studied RUNX1 and its role in establishing the blood system. So, my studies at that time were designed to inform methodologies for improving cord blood transplantation. But by the end, I felt still very isolated from, the cancer patients I had hoped to help. So when I finished with my PhD, I decided I needed to do something differently, something where I'm closer to patients. And I joined the Prostate Cancer Foundation. And there I worked directly under the chief scientific officer and was really invigorated by the chance to work hand in hand with both scientists and patients and help together collaboratively shape how the foundation should fill funding gaps in discovering new prostate cancer medicines. But after several years there, I still was missing something. And what I was missing was that I didn't know, how do you actually make a medicine? So I left PCF and I joined the biotech company Amgen. And I spent 8 years there working in various roles of research and development. And right about the time when I met Monica and Tim was around the same time that I was beginning to feel frustrated by the really incremental advances we were making in cancer therapeutics. So each new drug that gets approved, we get really excited about the advancements in cancer and oncology, but if you really look at the science and the data, we're only adding a few extra weeks to a patient's overall survival. And so I began to really understand that the only way we were going to beat cancer was to catch it as early as possible when a cancer cell is not even quite a cancer cell, it's pre-malignant, or at least when it's the very first few cancer cells, and treat it as soon as possible. So I was really inspired by the Babich's goal to pursue cancer interception and cancer prevention strategies. I felt that with my wide-ranging professional experiences, including having studied RUNX1 and understanding the blood system, that together that I just might be able to help them. And I have to also say that right off the bat, when I met Monica and Tim, I knew that this was a power couple that would get things done. And I certainly wanted to align myself to people like that.
Tim, how did you set out to make RRP different from other rare disease nonprofit organizations?
Yeah, well, Alex, a lot of it was what I'd spoken about a little bit earlier, where we're extraordinarily mission driven, and everything that we do comes back to that mission, which is to intercept RUNX1 FPD before it progresses to full-blown AML. So, we have multi-year strategic plans, we've got annual plans, everything sort of ties back into that. So we want to make sure that everything that we're doing is in support of that mission and aligned with that mission on there. I think another thing that's really been important to us that we try to, again, which I think we might do a little bit differently, is we try to borrow from successes of other organizations. So we're always trying to understand what maybe other nonprofit cancer prevention or cancer research organizations are doing, or maybe biotech or pharma in certain other diseases, and see, like, is it applicable to leukemia research? Is it applicable to cancer prevention? And so we're always trying to sort of see what other people are doing and not try to reinvent the wheel, because we can leverage the dollars and research that they've done. And I guess lastly, something I talked about was, we always try to be very, very collaborative. That's something that, oftentimes we see people doing research in just the broad academic scientific community in silos. And that's something that is very important for us, both sort of to ensure that in our grants that doesn't happen, but also sort of through, Katrin's working with people and encouraging them to collaborate, and, in ensuring that people feel like there's, conversations and collaboration is mutually beneficial. So that's kind of how we think about, what we're trying to do a little bit differently.
Wonderful. Thanks, Tim. Katrin, on that same note, what are the core values you think have made RRP so successful in such a short time?
Well, I think my answer will probably overlap quite a bit with what Tim just said, but I would say that there are really three core values at RRP that have made us effective. The first is focus. As you can imagine, when there's very little known about a disease, there are lots of good ideas and many different directions one could go. But at RRP, we are really laser focused and extremely disciplined about how we spend our time, and of course, how we spend donor dollars. So we make it abundantly clear to anyone and everyone who will talk to us that we're interested in really only one thing, and that is pursuing the discovery of a treatment that can either intercept at very, very early stages of cancer or even prevent cancer. And so, this takes a lot of discipline. And as Tim mentioned, we're constantly doing strategic planning, and it requires us to regularly review the scientific landscape with our SAB. It requires us to regularly reach out to our patient community, ask them about, their questions. What do they think is missing? We have a patient data hub, which helps us collect symptoms from patients. All this is really important in helping us maintain, again, that focus and make sure that we're, as Tim alluded to earlier, asking the right questions and knowing, where's the technology today? How quickly is it advancing? And helping us know where we should say no sometimes, or no, not right now.
Mm-hmm.
That's one of our core values. I think I'm not going to go into collaboration because I think Tim spoke to it very nicely, that collaboration is just a cornerstone, I think, to success in any industry, but I think most especially in science and medicine. And I would say the last core value is data sharing. I think that getting people to work together is one step, but then actually getting people to share data regularly is another really important piece of that collaboration. Data sharing helps you make sure that everyone is learning together as quickly as possible as one cohesive group. And it also helps you make sure that you're avoiding any redundancies across groups and teams, and hopefully learning from failures as quickly as possible. So I would say that those are our core values.
Tim and Katrin, I want to ask you both. I'd love to know what achievements you are most proud of. And I guess we could start with you, Tim.
Thanks, Alex. I think the first thing that comes to mind is really our partnerships with other organizations. I think that, you know, we've been very keen to find other like-minded nonprofits in order to figure out how we can work together towards cancer prevention. And I give a lot of credit to Katrin to being a really good partner with those organizations, so they feel that it's been, you know, beneficial to both organizations. Just to give a few examples, you know, the Chan Zuckerberg Foundation, the PCORI, you know, which is part of the federal government, the Mark Foundation, Alex's Lemonade Stand. We've co-funded grants with all of them, and or received grants from all of them, and worked together with them. And so I think that, again, you know, it's creating a multiplier effect when you're able to work with other organizations effectively. I guess the second thing that really comes to mind, maybe the most importantly, is you know, we have a really dedicated patient community and family and group of families. When we started the foundation, we didn't know any other families that were out there. And you know, the first individual I was introduced to is actually on this call here, is Sean Cryan. And I went and had dinner with Sean and his wife in Seattle years and years ago. And you know, it's hard to believe that there's several hundred individuals now in the community, and that's really a testament to all the hard work of you know, Amanda, Egan, and Katrin, and working to build out the community and foster a sense of community in here. So that, to me, that's sort of the second and maybe the most impactful achievement, I think, of the foundation since we started.
Great, thank you. Katrin?
I agree with everything that Tim said. I would add that I'm very proud of our ability to move from the lab bench into the clinic so quickly. In fact, we just got word yesterday that the very first clinical trial testing a drug called imatinib for RUNX1 FPD patients will officially open. And in addition to that particular clinical trial, there are 2 more clinical trials launching in 2024. And the reason I'm so proud of this is because these achievements prove that we can move a discovery that's made in the lab into a trial in just a few short years. Plus, I should also highlight that the discoveries and the ideas for these 3 clinical trials all came from RRP funded projects and our scientific convenings. So, you know, none of this would have been possible if we didn't have such a committed collaborative research network. And again, if we didn't maintain that laser focus on our goal. I also want to highlight that one of the reasons we've been successful in moving things from bench to bedside now so quickly is because we've leaned into a strategy called drug repurposing. And so what we've done is we've prioritized funding projects that focus on identifying and testing drugs that you can already grab off the shelf. It's already been approved. You can find these medicines in a pharmacy. And so why is this helpful? You know, the first reason is it's much faster to move from the lab bench to bedside in this setting because certain regulatory required studies have already been conducted for these drugs because they've already been approved. The second is that it's cheaper because you can actually skip those additional studies required by regulatory agencies. And also the drug is already being manufactured. So you win there, you have a huge amount of cost savings. And then the third reason drug repurposing makes a lot of sense, especially for a rare disease like ours, is that you don't need to go through a full regulatory review, which also, by the way, costs quite a bit of money. And that's beneficial because we can design these studies to not have to run for multiple years. And that's because if we had to go through the FDA or the EMA, the studies would require us to measure, well, how many patients actually develop a blood cancer. Not having to go to regulatory agencies allows us to design studies using surrogate endpoints that aren't yet what we call validated. So really 3 reasons why we go after drug repurposing, and it's helping us increase our speed towards our goal.
Great. Katrin, again, how do you see our funded research impacting other diseases outside of RUNX1 FPD?
Well, today we know that if a person is born with a mutation in one of probably over 40 genes, their risk of blood cancer is significantly increased. So RUNX1 is only one of those genes. There's a whole list of genes. And interestingly, that list is continuously growing. In fact, a new gene was just discovered at and announced at the conference I was just at last week. So going back to what I said in the beginning, when I said that I was really inspired by the Babich's courage to go after cancer prevention, because I believe, and so do they, that this is really the only way we can bend the curve on cancer death rates. I think that our collaborative community is really poised to deliver a safe blood cancer intervention for RUNX1 FPD. And when we do, it will represent a major proof of concept that, other cancer prevention, other cancer predisposition syndromes can also benefit from. I think I would also add that because genetic sequencing is occurring more and more for a variety of different health reasons, medical providers are detecting pre-malignant blood cells more and more in the general population. And so one can imagine that if RUNX1 FPD patients are benefiting from a treatment that directly targets pre-malignant blood cancer cells in their bodies, it's quite possible that people in the general population who may not have a genetic predisposition for blood cancer, but have evidence of these pre-malignant blood cancer cells in their blood, they could benefit too. So to me, the possibilities are really vast, and I'm truly energized by that.
What research, and other developments are you most excited about for the future?
I'm excited about, of course, the clinical trials that I mentioned and those that are launching in 2024, because we're going to learn a lot from these trials, and hopefully we'll be well on our way to finding a treatment that can actually reduce blood cancer risk. But I have to say, having been in industry and experiencing firsthand that drugs often do fail, in fact, 9 times out of 10, I know that it's quite possible that these first trials may not lead us to a cancer prevention. So I'm excited about all of the other projects that we're funding. So in parallel, we continue to fund really critical basic and translational projects that are homing in on the exact molecular and cellular mechanisms that are altered by RUNX1 mutations. Because today, we don't understand yet exactly how RUNX1 mutations are increasing blood cancer risk. We don't know why some individuals go on to develop a blood cancer and why others do not. And so if we learn, for example, in one of these trials that are about to launch, that it doesn't work, well, then the hope is that as we in parallel are making advances in our ongoing fundamental science projects, we can help understand why something didn't work. And on the flip side, also generate new ideas, new potential therapeutic approaches for cancer prevention. So I guess what I'm saying is I'm excited about the fact that we have a strong research strategy, a strong approach where we've really diversified our research funding so that we're making sure that we're learning through the fundamental science and also pushing it forward and making bets with some of these early clinical trials.
Great. And then, finally, Tim, where would you like to see RRP in 5 years?
Yeah. Alex, generally speaking, people talk about from the bench to the bedside in cancer research or any kind of drug research. And so we'd like to see it progress a lot more from the bench to the bedside. And so what are those things that matter in there? I think it's developing a greater clinical and clinician network globally, both in order to identify more patients and have patients come into the community, And this enables us to magnify the benefits of any kind of research that we're funding or any kind of therapies that we're able to identify, because it's going to impact more people. So I think just creating that bigger network of clinicians and patients on a global basis is number one. What that also is going to allow you to do is to really get an understanding of who's at risk of cancer on there, because we're getting a better sense, but we really don't know specifically what are those predictive factors for why someone is at risk and when. And then when that leads on to like, what is the timing of an intervention? Because if you can actually find out who's at risk and when they're at risk, then you can actually make a decision about what kind of therapy that people could take, or people could make their own decision about that, because they'll be informed about just, the likelihood of progressing to cancer and how serious or not it is, because you don't want to just take a drug or a therapy for the sake of it. You only want to have to take a drug or a therapy if to the extent that you feel like there's a high probability in the immediate future that you're going to progress to cancer. And so that's our goal. Our goal is really to have therapies and to have people be informed about what is the appropriate time in their lives to be able to take those therapies.
Fantastic. Thank you so much, Tim. And thank you, Katrin. I think, this is exciting developments that we've had in a short time. And I just really hope everybody enjoyed the conversation we had today. As we're kind of getting close to the close of the event, I wanted to open it up for any questions that you may have. So again, you can write it to the group in the chat, or please just unmute and ask your question directly. Great. Well,
I do have one.
Oh yeah, Sean, I'm sorry, I didn't see.
I'm curious for Katrin or Tim, if either of you have seen any other groups trying to adopt the same methodology, trying to increase their hit rate, or their whatever you want to call it, the speed with which they can take possible treatments to the bedside, I guess.
Katrin, do you want to take that one?
Yeah, sure. I can. This is certainly a hot topic in the rare disease community because it's so costly to develop a new drug, right? And because we need, as I mentioned briefly, validated endpoints, and so there's a lot of challenges. So drug repurposing is something that others have tried and have been successful at doing. And one very famous example, if maybe famous to me just because I'm in the space, but there's an individual actually at UPenn, his name is Dr. David Feigenbaum, and he has discovered a treatment for his own disease, a rare disease called Castleman's disease. And that particular treatment is a treatment that he found, that's available in the pharmacy, that he's basically saved his life with. And he did this through doing his own lab bench science at UPenn while hoping to stay alive. He had a very terminal, the condition is quite terminal. So he's written a book about this whole process. But yeah, so there are examples. And as Tim mentioned, we try our hardest to make sure that we're not reinventing the wheel. And if something has worked, let's implement it. Thanks for the question.
Fantastic. So we have another question from Taylor. For the clinical trials, how are participants identified, chosen?
Such a good question. So for every clinical trial, there's always a list of inclusion criteria and exclusion criteria. And that is defined by both the principal investigators, the people who are leading the study team, but also in collaboration with a third neutral party called the Internal Review Board, which usually sits at an institution, or it can be outside of an institution. And they're responsible for making sure that every clinical trial is designed as safely as possible so that patients aren't, as best as possible, aren't harmed. And so the criteria are defined by those two expert groups. And in order to find out whether you're potentially eligible is just by reaching out to the study team. So we have this imatinib trial that I mentioned, for example. I think in the newsletter today, you can find information about the study, and then come January, we'll send out information for those other trials as well. And so there'll be a contact name. You can reach out, and then they will basically have conversations with you and let you know whether you're eligible or not. But the hope is that many, many people can be eligible for these studies. That is the intent.
Great, thank you, Katrin. And thank you, Amanda. I know that you're putting some information on the clinical trials. And I just wanted to allow for any other questions before we close for today. Great. Well, this has been a wonderful conversation. I want to thank Tim and Katrin for their thoughtful answers. I want to thank everybody for joining us today and for this wonderful conversation. I want to say how appreciative I am and the rest of the RRP team is for your support. As you know, we couldn't advance the science and impact patients and their families without your support. And so if you haven't already done so, please consider providing a gift before the end of the year during this holiday season. All donations will be matched 1-to-1, doubling your impact. And you can, of course, give any time at RUNX1 FPD. Again, thank you all for attending, and I wish you the very best this holiday season. Thank you again. Take care.